Hello, everyone. Welcome back after the session break. Continuing with today's schedule, it is my great pleasure to present to you our next speaker, Dr. Claire Deo. Claire did her PhD at the École Normale Supérieure in Paris, France, working on light-responsive molecules. Pursuing her interest in this field, Claire joined the Howard Hughes Medical Institute as a postdoctoral fellow. She is currently a group leader at the EMBL Cell Biology and Biophysics unit at the University of Heidelberg. The focus of her group is developing novel fluorescent probes for advanced biological imaging. With this, I would like to invite Claire to our virtual stage. Claire, we are very much looking forward to your talk.
Thank you very much, Margarita, and thank you for the introduction. And also thank you again to all the organizers for putting together such a nice program and managing the organization. And it's my pleasure to be speaking here today. So I'll just put the pointer. Yeah. So I am a chemist, and what I'm interested in is how we can use the knowledge of chemistry to build and tailor molecules that are specifically designed for biological applications. And in particular, one of my main interests is the design of fluorescent reporters for biological imaging. So today, I want to give you a short introduction on fluorescence and fluorescent molecules, and give you a little bit of an overview of how we can label molecules and new methodology, and show you a couple of different projects that I have been working on in the past in these directions. So what is fluorescence? So fluorescence is the process of emission of light following absorption of light. When a molecule is illuminated at the wavelengths at which it absorbs light, it reaches an excited state, and then it can then release this energy, and if it releases by emitting light at a different wavelengths, it's called fluorescent. There's different parameters that we use to quantify the efficiency and the characteristic of the fluorescent process. So the absorption and emission wavelengths are what characterizes the color of the fluorescence. The extinction coefficient is a measure of the efficiency of the absorption process. It quantifies how strongly a molecule absorbs. And the quantum yield is the number of the photons emitted over the number of photons absorbed. And this is a metric of the efficiency of the fluorescent process. So for the fluorescence to be highly efficient, you want this value to be as close to 1 as possible. And together, the efficiency of the absorption and the efficiency of the emission is what characterizes the brightness of fluorescent reporters. There's a very large diversity of molecules that are fluorescent. Most of the ones that are used in biological imaging are composed either on small molecules and or fluorescent proteins. So historically, the first fluorescent compounds were small molecules. The very first compound for which it was observed is quinine. Quinine is actually an antimalarial drug, which is why it had attracted a strong interest. It is extracted from a tree. And you're also all familiar with it because it is the molecule that gives the bitter flavor in tonic water. And this is also the reason why your gin and tonic might look like this in a nightclub. So this process of fluorescence was actually observed for the very first time 200 years ago, and was characterized by blue glow in the sunlight of a solution of quinine. It was a few years later that it was really identified that fluorescence was actually a change of the wavelengths of light when it was going through this solution. And this was demonstrated by Gabriel Stokes through a very smart experiment where he illuminated a solution of quinine with sunlight. And then by looking at the light emitted and blocking with a yellow filter any incident light, he could observe really that this was a complete change in the wavelengths. And the beginning of the synthesis of small fluorescent molecule was in 1856, and actually started with the first attempt to synthesize quinine, because as an antimalarial drug, this drug attracted a lot of interest, and instead of extracting it from a tree, people started wondering if we could actually make it by utilizing coal tar, which is a byproduct of the coal industry. And this was the task of William Henry Perkin as a PhD to synthesize quinine. And this completely failed and led to this compound, which is called movane, and is actually the very first synthetically made dye. And that was the birth of the dye industry. And starting from this point, many chemists started synthesizing this highly aromatic scaffold and studying their fluorescent properties. And this led to most of the fluorescent compounds that we know to date. And this is just a small overview of the diversity of chemical scaffold that you can find in fluorescent molecules. Most of these things can be obtained commercially now, and you might be familiar with many of them, such as DAPI or HUSHT, which are used to label DNA, the scaffold of fluorescein and tetramethylrhodamine, and then also, for example, the cyanine dyes with Cy3, Cy5, Cy7. And this diversity allows to cover the entire spectral range, leading to a very diverse palette of fluorophores, but also an extensive knowledge on how, by introducing small variation on the chemical scaffold, we could affect and modulate the fluorescent property. And early on, the process of fluorescence attracted interest as a potential imaging modality, and there is many reasons for that. First, because fluorescence, as it can in principle have a very high contrast because most biological molecules are non-fluorescent. It is highly sensitive, can be seen at the single molecule level. It is a very fast process. The fluorescence emission in itself is in the range of a picosecond. It is minimally invasive because light can go through tissue. The diversity of colors enables easy multiplexing. And finally, fluorescence is a process that is environment sensitive, which can allow to monitor different environment. And all this has led fluorescent microscopy to emerge as the main modality to monitor the structure and the dynamics of biological systems today. So going back to fluorophores used for biological imaging. So small molecules have the advantage of being very bright and photostable, and as well as giving access to a wide range of structural diversity. However, they have a main caveat, which is the labeling specificity that is often unpredictable and uncontrollable. If you deliver a small molecule as is on a biological system such as cells, it is often impossible to predict which features will this compound label and how selectively. So initially, the first approach to use these dyes in fluorescence imaging relied on the methods to make reactive fluorophore by attaching a highly reactive group to common fluorophore scaffold. So this led, for example, to FIT-C or compounds such as Tamara SE. And the way this compound work is that these groups react very rapidly with free amines or thiol groups that are present in proteins or nucleic acid. But they're not selective, so they would react with all the groups in principle. So this is very useful to label isolated protein or isolated nucleic acid, and then can be delivered into cells. And this is what led the way to methods such as immunofluorescence or FISH, but they're not applicable to live cell or organism imaging. And things changed dramatically in the late 90s with the discovery of fluorescent protein, with GFP that was isolated and identified from the jellyfish Icora victoria. And this compound is constitutively fluorescent based on a sequence of cyclization and followed by oxidation to form a fluorescent chromophore. What made this success is the ability to express green fluorescent protein as a fusion of any given protein of interest. And this led to the Nobel Prize in Chemistry in 2008. Their ability to create transgenic line was then followed by a lot of research to expand the spectrum of fluorescent protein, which can now cover a whole different range of colors. And the emergence of fluorescent protein actually completely shifted the field of fluorescence imaging, enabling imaging in living cells and in vivo with very high genetic specificity. But fluorescent proteins also have their limitation. In particular, they're not as bright and photostable that what can be achieved with small molecule. And to illustrate this, This is a graph showing all existing monomeric fluorescent protein ranged by brightness as a function of their emission wavelengths. And what we can see is that there are some pretty good fluorescent proteins that perform in the green. However, their brightness drops extremely fast as we move to the longer region of the spectrum. And in contrast, this is just a subset of commercially available synthetic fluorophores. And even in the green and yellow area of the spectrum, they clearly outperform fluorescent protein. And this becomes critical in the red region, where you can actually buy fluorophores that emit at wavelengths that cannot be achieved with the fluorescent protein. So to circumvent the limitation of fluorescent proteins, that in addition to brightness and photostability also had pH sensitivity, their fairly large size, and their necessity for oxygen for maturation, hybrid approaches started to emerge that combine the advantages of both. So these approaches started emerging about 15 years ago with the appearance of self-labeling tags. Self-labeling tags are small protein tags that can be expressed also as fusion of a protein of interest, and that have the property to bind and recognize a small synthetic ligand in a highly specific way. And by attaching this ligand to a fluorophore, we can create this hybrid system that combines the genetic specificity of the protein scaffold with the brightness, photostability, and molecular diversity of small molecule dyes. The very first example was the SNAP tag that was developed in 2003, which is a 20 kilodalton protein that recognizes this small benzoguanine ligand. And then later on with the HALO tag, which is slightly bigger and binds covalently to this chloroalkane group. So the ligand is in principle generic. So we can attach any group we'd like to the ligand, such as fluorophore or other handles for bioconjugations or other type of labeling. And this approach really prompted the reemergence of the interest in the field of small molecules for fluorescence imaging. So they're mostly used with the scaffold of rhodamines, and there are several reasons for that. First, rhodamines are fairly bright molecules. They're cell permanent, and they have a diversity of different positions that can be very easily functionalized. By tuning the structure, we can access to a whole different range of color. And rhodamine also have the interesting property that they exist in equilibrium between a closed form that is non-fluorescent and an open form that is brightly fluorescent. And this equilibrium is environment sensitive and can be controlled, which has also led to the implementation of a variety of functionalities into self-labeling tags. So for example, some fluorescent rhodamine ligands are fluorogenic, meaning that they show fluorescence turn on when they bind to their tag target. This was found initially on the compound, this silicon rhodamine compound, which is a farad fluorophore. But in solution, the ligand is almost non-fluorescent. However, when it binds to the halotag protein, it was observed that the equilibrium is actually shifted towards the more open form. And this can be observed with an increase in fluorescence about 7-fold. This can be rationalized because the fluorophore, when bound to the halotag, sits in very close association with the protein scaffold. So by changing the dye microenvironment, this equilibrium is shifted. And this equilibrium can be rationally modulated by introducing a variety of substituents to create compounds that are more and more fluorogenic. So starting from silicon rhodamine that showed 7-fold, if we introduce this electron withdrawing fluorine, we get a fluorescence increase about 100-fold. And then if we introduce some other electron withdrawing group on the bottom ring, we can reach further increases in fluorogenicity with this, exemplified with this orange compound that shows an increase of fluorescence of 1,000 upon binding. This is particularly useful for imaging because you can realize imaging without washing and providing a very low background. So as a comparison on the left, you have a ligand that is not very fluorogenic. What you can see is that it does label the nuclei where the halotag is expressed, but you also see a lot of signal in other areas of the cells. In comparison, if you use a very fluorogenic ligand, every molecule that is not bound to the halo tag will remain dark, and you can then image with a reduced background. So overall, the self-labeling tags used in conjunction with rhodamine have emerged as a very robust labeling platform. They're bright, photostable, multicolor, very tunable, and combine the genetic specificity with all these properties. They can be used in living cells, they can be multiplexed with different tags, and the ligands can also be delivered in living animals, such as mouse, so they can be used for in vivo imaging. And what I've been interested in is, can we use all these interesting properties and re-engineer this self-labeling tag platforms into functional reporters that are able to report on dynamic events? Sorry. So what I've been in the past particularly interested in is to design sensors, fluorescent sensors for calcium. Calcium is a universal second messenger, and monitoring calcium is particularly interesting in the field of neuroscience because it serves as an excellent proxy for monitoring neural activity. The gold standard for calcium imaging is the sensor HaleCalMP, which is a genetically encoded scaffold based on a green fluorescent protein and a calcium binding domain. And HaleCalMP is a very good sensor. It is fairly bright, shows a very high fluorescent variation in response to calcium with a 62-fold increase. But efforts that have been made to design more redshifted versions of a calcium sensor result often in limited brightness and much reduced sensitivity. So for example, platforms that emit in the orange have limited brightness and much lower sensitivity. And one of the only existing sensor in the far red is negative and is much dimmer than HaleCalMP. So we thought to design new calcium sensors based not on fluorescent protein, but on rhodamine and tag, as in the far red, rhodamine can be significantly brighter than fluorescent protein. And this was work that I realized during my postdoc at Ginelia in the group of Luc Leives and in collaboration with Eric Schreider. And we worked on two different approaches for the generation of calcium sensors based on rhodamine tags. And the two approaches differ in the sense that in the first approach, we used a synthetic calcium sensing domain, and in the second approach, a genetically encoded sensing domain. So in this first approach, we first sought out to design a synthetic calcium indicator and combine it with the halo tag. So synthetic calcium indicators are all essentially based on this BAPTA, which is a calcium chelating moiety. And in the calcium-free state, electrons transferred to the fluorophore quenches all the fluorescence. But upon binding to calcium, this effect is suppressed and we recover fluorescence from the fluorophores. So to design a new calcium sensor, we started with the rhodamine fluorophore and sought to introduce the calcium binding moiety at different positions. And we first started by introducing it on the bottom ring, and then further on the top small rings. And we've systematically characterized how the relative position of the two groups affected both the quantum yield and the brightness of the sensor, and also the sensitivity and response to calcium. And we found that this configuration was optimal, retaining a very high brightness and with a fluorescence increase upon binding calcium of about 17-fold. So next, we tested this compound in cells to see how they perform. Here you can see in vitro the increase in fluorescence as calcium increases, but is this conserved in cells? So if we deliver this compound into cultured neurons, what we can see is that it labels preferentially the nucleus. So this really further illustrates the challenge of working with fully synthetic molecules, in which we cannot really predict where they're gonna localize, and we have no control over it. So to be able to genetically localize this sensor, we combined it with the halo tag ligand, as I shown before. We tested that the calcium response didn't change, whether it was bound to the protein or not bound to the protein. And then we tested it into cultured neurons as well. So this time, to really demonstrate that we could invert the standard localization of the indicator using this method, we expressed a nuclear excluded halo tag. So here you can see on the cell body that the nucleus is dark and that the cell body is bright. So that means that this molecule labels efficiently on the halo tag, and that we can go around the natural localization using this method. To monitor how it responds to calcium, we can stimulate the cells electrically, which trigger calcium fluctuations, and we can measure the fluorescence. So the sensor responds well in cultured neurons. And it is about 5 to 10 times brighter than the two existing fluorescent protein-based sensors that work at the same wavelengths. We then extended this approach to far-red fluorophore only by changing the core rhodamine with the silicon rhodamine. And this compound performs in the far-red similarly. And we've demonstrated that this is useful to monitor calcium in very small compartments by monitoring calcium fluctuation in primary cilium. And this compound shows an increase in fluorescence of about 60% when calcium is uncaged. So this approach showed that we could use a synthetic domain to design tag-based calcium sensor that perform well in the far-red. But there's still some limitations. So first, the molecule that you have to deliver is fairly large, which can pose problem if you want to deliver them in vivo and in animals. Making them is fairly complicated, and also the sensor is fully functional even when unbound. So if the small molecule mislocalizes, you can still have some background issue. So to circumvent this problem, we were working on a second approach in which the calcium sensing domain is this time part of the protein scaffold. So this has two advantages. First, we can deliver much smaller molecules. And secondly, as the sensor is separated itself into two parts, the sensor becomes functional only when the fluorophore is bound to its target. And this abolishes problem of potential background from the ligand. To design the sensor, we reasoned on the open-close equilibrium of the rhodamines that is environment sensitive and that I've presented before. And we designed the scaffold in parallel with the design of the GCAMP calcium sensor. So GCAMP is, as I mentioned, a GFP derivative bound to a calcium binding domain, which is composed of calmodulin and a peptide. And in the presence of calcium, these two domains assemble. And this triggers a very large change of conformation in the close proximity to the GFP chromophore, which results in a change in fluorescence. So we reasoned that we could substitute GFP in this design with the rhodamine halo tag, because we know that the rhodamine in itself is also an environment-sensitive fluorophore. So we call this design HaleCalMP in comparison to GCAMP based on halo tag. And we started by engineering the protein in observing the crystal structure of the halo tag, and with the reasoning that we wanted to introduce the calcium binding part in very close proximity to the fluorophore to maximize the interaction between the two. So we introduced the calcium binding part at this position, and this led to the design of two variants of the first calcium sensor based on two different peptides. And here you can see the crystal structure. So the calcium bound domain is very similar to the GCAM part. And here you can see the halo tag with the far-red rhodamine sitting on the cavity of the halo tag. So we tested the response, the fluorescence response upon addition of calcium of our design. And we can see here that we see a large increase in absorption and in fluorescence. With our two variants, one was brighter with a lower sensitivity and one was slightly dimmer, but with a higher sensitivity. One benefit of this design is that we have access to these far-red wavelengths that are not really accessible with fluorescent protein. But moving to this two-part approach, one advantage is that the scaffold becomes highly modular by varying only the small molecule that is delivered and conserving the same protein scaffold. So we can, for example, change on demand the color of the sensor by changing the small molecule dyes that we deliver. So we can, with the same protein, have an orange sensor, a far-red sensor, or a green performing sensor. We can also fine-tune the properties of the sensor and while keeping the same wavelengths. So by changing slightly only the open-close equilibrium of the rhodamine by introducing different substituents on the azacitidine group, we conserve the farad emission, but we can alter the brightness, the dynamic range, and also the affinity for calcium of the sensor. So, for example, this was the original sensor. We can generate a whole family of different sensors. Some of them showing, some of them being dimmer, but with more reduced calcium-free fluorescence, and some of them may be less sensitive but brighter. And those can have applicability depending on different experiments. For example, when looking at very small and sparse features in a system, it might be beneficial to have a higher background to be able to unambiguously identify this feature prior to monitoring calcium. At the opposite, if we're looking at large volumes in which the sensitivity becomes more important, might be more interesting in using this small molecules. So the platform is really highly tunable, which is also something that cannot be achieved easily by engineering fluorescent proteins. So finally, we tested this platform in cultured neurons, similarly as we did for our other sensor. So we expressed the HaleCalMP platform as a nuclear excluded sensor again, and then delivered the small molecule dye. And we can quantify the fluorescence response. We can quantify the fluorescence response in response to the different electrical pulses to benchmark the sensor. So you can see here the nice fluorescence increase upon electrical stimulation. So with our first fluorinated dye, we get about 200% delta F over F upon stimulation with 160 action potential. The fine-tuning property using different small molecules can be conserved in cells. So by using different dyes, we obtain different dynamic range. And as a point of comparison, this is how NearGecko performs in the same condition. So NearGecko is based on this near-infrared fluorescent protein. So our sensor is significantly more sensitive. And we can also measure the kinetics of the sensor, which is the fluorescence, the on-fluorescence response and the off-fluorescence response. Different variants of our sensor are also faster than NearGecko. So to conclude, we've shown that we can use fluorophore type pairs as a substitute to fluorescent proteins in biosensors, that this allows to overcome certain limitations of fluorescent protein-based reporter, such as access to farad wavelengths with bright and fast sensor. This sensor is importantly highly modular without having to re-engineer the sensor platform. And in addition, this sensor is also generalizable. We can imagine that, so this was exemplified with calcium, but that this can be applied and transferred to many other small molecules, ions, and analytes for which they are identified genetically encoded binding motif. So with this, I would like to finish by thanking everyone at Genelia in particular that contributed to the projects that I have presented today. And I also like to thank my new group at MBEL. And thank you very much for your attention.
Thank you very much, Claire, for this amazing talk. Now we have quite a bit of questions. I will quickly go through the most upvoted ones. So the first question is from Grasvidas. Why signal to background of fluorogenic halotag ligands is very different in test tube and in living cells?
Yeah, that is definitely something that we observed, that although the range is usually conserved, that a less fluorogenic dye in vitro will be also less fluorogenic in cells. There is still some background that is observed, and this can be due as still certain non-specific interaction that I guess can probably still activate the open-close equilibrium, even without it being really halotag bind driven. But yeah, there's definitely something with the snap tag, for example. What we observe in cells cannot always be reproduced in vitro and vice versa.
Okay. The next question is actually from me. How specific are the ligand tag reactions? Are they temperature sensitive, and how can the efficiency of these reactions be increased?
So they're very specific, in particular with the SnapTag and HaloTag. They're very specific, which is what makes their success overall. They don't have any endogenous ligand, which is great, meaning that they're orthogonal to the biological system. It is temperature dependent, all binding. The binding of Snap and Halo is as kinetics that are sufficient to work well in cells and in systems. So when you deliver the ligand, you usually see the fluorescence turn on in a few minutes. And after incubating for 30 minutes, you usually have saturation of the protein. And I'm sorry, what was the last part of your question?
How can the efficiency of the reaction be increased? Yeah.
Yeah, so there's been a lot of effort towards this in introducing some specific point mutation in the protein tag scaffold, and this has led to the current versions that we use that have been optimized for this binding kinetic. In terms of the efficiency for both these tags, the labeling is covalent, which means that in principle, as long as you add a sufficient amount of the ligand, you will saturate, so you will reach the maximum labeling state.
Okay. Is it possible that there is excess of the ligand or the tag remaining and that could block the signal in the cells, for example, when you're labeling the cells? Is it possible to overcome this issue?
Yeah, so for excess of the ligand, this is what we usually do in principle. We add an excess of ligand, wait a little bit until all the protein is marked, and then you can wash out the excess of ligand. As it is cell permanent, you can also wash it out. So you can remove the excess ligand. Using those fluorogenic dyes, one of the advantages is that even if you have excess and you still have ligand floating around, they will not give too much background. Although, as we've seen with the previous question, you can still see some non-specific things happen.
Okay. Thank you very much. Okay, so our next question is from Daniel. What is the best resolution achievable with the Snap or Halo tags, given their presumably quite large size and thus offset from actual protein of interest?
Yeah, that is absolutely right, which is that the fluorophore is very small, but you have to use a fairly large tag to label it. So Snap tag is slightly smaller than the fluorescent protein. Halo tag is slightly bigger, 30% bigger. So that does introduce some labeling error, and that becomes an issue when you move to super resolution techniques because then you can see this offset. So yeah, it is important to know that it's there. It's not as bad as using antibodies in terms of what you get in terms of labeling errors. But the barrel of the Halo tag is still a few nanometers large. So there is this kind of shifting.
So you can still detect the shift.
You have to know that it's there because this will be an artifact of what you observe, which is if you're looking at really super resolution of very fine structure, you have this extra layer added by the label on the sides.
Okay, thank you. So we have time for a few more questions. So the next question is from Nora. How do you deliver the fluorescent compounds into living cells or in vivo? Are they membrane permeable?
Yes, so most, you can tune that, permanent versus non-permanent, but rhodamines are cell permeable. So the way you can deliver them is fairly simple. So you just add the ligand to the cell culture medium, you incubate, and the ligand will cross the cell membrane and find its target. Then you can wash out the excess. For animals, it can be a bit more complex depending on what you want to label. In what I presented, for example, with applications mostly for neuroscience, so for labeling the brain of living animals and full awake animal. One thing that has also made the success of this rhodamine Halo tag ligands is that they have the ability to cross the blood-brain barrier fairly easily. So for zebrafish, you can add it to the zebrafish water, have the zebrafish swim in it, and the dye will label the brain. For flies, you can put them in the fly food. For mouse, you can do a bloodstream injection, and the dye will cross the blood-brain barrier and label the brain. So it's still fairly simple to use, but the efficiency of this can depend on the structure as well.
Okay, thank you. So next question is again from Grasvedas. Does binding of calcium influence HaleCalMP reaction with fluorophore substrate?
Yes, absolutely, and that has been actually in the beginning of the development of a project, one issue that we had, which led to substantial artifacts in assays, because what happened was that when the sensor was calcium bound, the binding to the fluorophore ligand was significantly faster. So when we were doing assays in which the ligand would be in excess, changing the calcium state would trigger more binding or less binding, and then introduce differences. So there is a difference, which in our platform I think is probably about a factor 2, and which is, I think, not that surprising, given that you have this very large change of conformation of the halo tag. I think that in the calcium-free state, maybe the protein pocket is disrupted, which slows down the kinetics of the ligand labeling. And upon binding calcium, it's more compact and more reconstituted, which facilitates the binding.
Okay. Yes, so next question from Sophie. How successful have halotags been in live imaging in the whole organisms, for example, in embryos?
So I mentioned already brain imaging for mice, for example, but it is used for Drosophila embryos or even mice embryos for labeling.
Great. Okay, so I think we still have time for 2 more questions, maybe. So the next question is from Tzu Chen. How does the chemical structure of fluorophore affect the dynamic range?
Yeah, so we don't know how exactly, and there's dynamic range. There is also the affinity for calcium, and there is also the kinetics. So this parameter seems to be strongly dependent on the fluorophore structure, which we don't explain, but it is definitely the case. I'm sorry, I think I misunderstood the question. The dynamic range, yes, we can explain actually, because the dynamic range is dependent on the open-close equilibrium, and this we can really fine-tune it. So if we use a fluorophore that is more on the closed form initially, then it will probably be dimmer in the off state, and then become a bit dimmer as well in the calcium state. And at the opposite, if we use a fluorophore that is already more open, then the range at which we can open it is smaller, so we'll have more background, but then it will be brighter. So this we can actually rationalize a little bit. What I was mentioning is in terms of is mostly affinity and kinetics of the sensor itself, that we don't understand yet how they are really fluorophore dependent.
All right. Thank you. I think we already went through the next question. So how to get rid of the excess amount of tag or the ligand so that it doesn't block our signal. And there is actually one question left. In cases where the number of calcium atoms involved in a process is very low, for example, sensing in cilia, do the sensors alter calcium dynamics and therefore the measurement of the phenomenon?
Yeah, that's a very good point, and I think that's always a critical question to ask, which is how much do you affect how your system works by observing it? And of course that has been observed before, that using calcium sensors, especially at high concentration, can result in a buffering of the calcium, and so you end up affecting the how your system would normally work. So it is important to keep in mind there is no real way around it except in doing controls and comparing different doing different controls, comparing indicators and seeing if the same behaviors are observed.
Okay, makes sense. Thank you so much. So with that, I would like to thank you, Claire, once again for the great talk and for being here with us today.
Thank you very
And
much. Thank you for the conference.
I would also like to thank all of our attendees for asking great questions. So with this, I would like to invite you to our next talk, which is also the last talk of our conference, which will be by Dr. Carissa Sanbonmatsu, and it will start in 15 minutes. Carissa will be talking about novel insights into the structure and function of long non-coding RNAs. So hopefully I'll see you all there. Thank you very much. Bye.